Yes, let's have a look at profile hidden Markov models. So this is all about sequence models, which means we try to describe a subset of sequences. So the data we start out with is always multiple sequences. And we can use BLAST to compare one sequence against another sequence, but that's really not what we are doing here. We are looking for gene families, which means we look at genes of same or similar function or sharing some common pattern. And these gene sequences, they vary a little bit across different genomes, of course, but they also have something in common. And that what they have in common is exactly what we are looking for now. So a sequence model describes sort of a population of sequences, and this population is a set of sequences who share this common pattern. So it describes some pattern typical for this population. Could be typically a gene family, right? Or a protein family. So this can be done in several ways. So let's focus on the profile hidden Markov model approach here now. And this is typically used when we have longer patterns. If you have very short patterns, motifs as we call them, we don't typically use this approach. We use other ways of doing that because short patterns are must also be very conserved. Otherwise, they are hardly patterns at all. But when you have very long patterns, you also have some deviations, more deviations from that pattern, right? And then this approach here is particularly well suited. It's used in several databases, and PFAM is the most famous one, I would say, describing patterns using these particular models. And the software hammer is what we use to work with these PHMMs, which we will do in module 7 of this course. So here is a typical start out. We have a multiple sequence alignment. We have many sequences, and we can see that these sequences, even though they are not exactly identical, they all share some pattern, right? There is something in common here. They all start with a T, for instance, and we can see there's also lots of A's inside here, and then they end typically with a T again, or perhaps a C now. Yeah. So they are different, but they are similar enough to be said to be containing the same pattern here now. So we use this as an example here now. And notice I use a DNA sequence example. This is not typical. This is most typical for protein sequences, amino acids, but I use DNA simply because it's smaller, simpler. The example gets smaller and everything is so much simpler. But we need to have a multiple alignment of this type here in order to sort of understand what this is all about. So we have a selection of sequences from this population. And the first thing we need to sort of tell ourselves is that these sequences here are probably just a small selection, right, of all the sequences that we could have had. So there are probably many more sequences that we have not yet seen, and we will now try to describe this pattern in such a way that when a new sequence comes along that also belongs to this pattern, we will sort of detect it. We will see that this new sequence also matches this pattern quite well. So we will try to build a model. And notice that the first thing we need to do is to actually do alignment. So first, in order to build a model, we need to do a multiple sequence alignment first. And there is no way around that. And this is really not optimal because all these multiple alignment softwares are suboptimal in the sense that they are all heuristics that tries to make the best possible multiple alignment, but They all cut corners in different ways, so none of them are guaranteed to find the sort of the correct multiple alignment. And for that reason, it's usually a good idea, if possible, to use several different software tools and see if they come up with very different results. If they do, it's really not a good sign. But if they do, try to sort of average over them in a sense, and always inspect alignments before you proceed. That's a sort of a good advice. And I mean, for the PFAM database, for instance, there's thousands of models there, right? And all of them must have had a multiple alignment like this. And there are actually people who are paid to sit and inspect this to some degree, to verify, curate, as we say, that the alignments are not completely way off. But of course, computers are better at this all the time, so I guess that it's a minimum of, what should I say, manual inspection required these days. And we should remember that any mistake we make here now will sort of pursue us forever after. It doesn't matter how good models we make afterwards if the sort of the alignment we started out with was rubbish. But okay, let's say this is not okay now. How do we proceed? Well, the first approach we usually think of is this profiles. What is a profile? A profile is simply a table of position-specific probabilities for the various symbols. So one row for each symbol in the alphabet, which means in our case 4 rows then, A, C, G, and T. One column for each position in the pattern, in the sequence alignment, right? And the columns are probabilities summing to 1, actually. So here is an example of such a profile. And we saw from the previous slide that that this had 8 positions, the multiple alignment. If you go back to the previous slide and count the number of symbols, you will see there were 8 positions. So that's why we have 8 columns here, and we have 4 different symbols, A, C, G, and T. And now this reflects the probability of the different symbols at the different positions. So when you see 1 here and 0 for all the other 3 symbols, it means that the first position is always a T. That's what this 1 down here means. While if you go to the second position, in the second position is almost always a T, but it could also be a C or a G. A few times, right? 5% probability, 90%. So this is the overwhelming, most likely symbol to occur in the second position. So T, T. And then we can see here is C, the most typical symbol, but A is also quite typical, right? But not G and T in the third position, and so on, right? It should be noted right away that we typically don't like zeros in such tables. We don't like zeros because a probability of 0 is sort of a too drastic statement. What it means is actually what we implicitly say here in the first column is that there can never be any sequence matching this pattern that has anything else but a T in the first position. Right? And that's too strong statement, because there could easily be sequences that we have not yet seen who matches perfectly in the other 7 positions, but who has an A or a C or a G here, right? So the probability should not be 0. It should be small, but not 0. So to avoid the zeros, we add what we call pseudo counts. This is not something I will dig into here now. You probably have heard of this before. You should have in BIN310 at least. So I will not dig into it, but I just mention it because zeros like this, it doesn't look good. Probabilities are never 0 or 1, right? They are close to 0 or close to 1, but not that drastic. Okay. So a sequence logo, that's a graphical display, and you may have seen some of these. This is a sequence logo. And this describes this pattern here that we describe by the profile numbers up here. We can display it graphically down here in this way. And you can see it's also 8 positions, right? 1, 2, 3, 4 along the x-axis here. And then we plot in each position the 4 symbols, in this case, with different heights, right? So in the first position, we only see a T and a very tall T, meaning that in the first position, you can only have a T, nothing else. In the second position, you'll most likely see a T, but you can also see C and G here. They are very, very small. In the third position, C or A is the typical ones, right? And so on. So this is a nice way of describing it. Why are the letters of different heights? Well, that has a very clear explanation. So let's look at the mathematics here behind this logos. Here we have the symbols of the alphabet. In our case, A, C, G, and T then. But in general, it's the alphabet of length Z, right? And then you can compute for each position, what we call the entropy. This is the entropy, and that's really these probabilities here are actually these numbers up here. These are the probabilities, right? In each position, in each column. So you take the probability for each symbol, multiply by log 2 of that same probability, right? And then we have what we call the information content of a column is simply log of the alphabet size minus the entropy. And what we plot on the y-axis over here is this information content multiplied by these probabilities again. So that means that when you have a very tall letter here, T and A, notice that they are the tallest and they are alone, right? They're always alone because then the entropy is at its lowest. When you have one symbol completely dominating in a position, the entropy is actually 0. There is no entropy left. There is no variation left, right? In these two columns here. That's why the T and the A here becomes big. When you have some entropy, the letters get smaller because you subtract something here, right? This information content gets smaller. And you see that's what we scale with down here. So sequence logos are graphical way of displaying exactly the same information we have here. It's a simple, straightforward formula. But let's go a little bit a step further, you could say. Profile hidden Markov model. So you can see again the profile word is part of this. What is this actually? It extends the profile idea to also include gaps. And that's important when you have long patterns, because the longer patterns you have, the more likely is that all the sequences that belong to this pattern, they are not exactly same length. There are some gaps in here and there to compensate for the variable lengths of these sequences. They can be 100 symbols on average, perhaps, but some are 101, some are 98, some are 104. It varies a little bit, right? So in order to describe this, we need to include some gaps. And that's the big advantage of hidden Markov models. We can introduce these gaps in a very elegant and nice way using probabilities. So let's start out by considering a case without gaps then, because that's the easiest way of describing a hidden Markov model. Each position in the pattern is represented by what we call a hidden state. And from each hidden state, symbols are emitted by different probabilities. So what on earth does this mean? We will see. And there is some probability of going from one state to the other state. And that's where the Markov modeling comes into place. But for now, these probabilities are 1 all the way through. Let's have a look. Here we have the first hidden state, M1. Then there is a certain probability of moving from M1 into M2, and since there are no other arrows, at least by now, there are no, so if you are in this state, you have to go over here because there are no other paths, right? And from this state, you can move further along the arrows, but there is only one arrow, so you have to go to this state, right? And so on, and so on, and so on. And now we have, yeah, I think perhaps even one more, 8 of these hidden states, because we had 8 positions, right? Remember, we had 8 positions in our multiple alignment, and then we would typically start out like this. 8 different hidden states, and from each of these hidden states, symbols are emitted. Yeah, and here the transition probabilities is 1.0, simply because there are no other options, right? How can you move out of this box? Well, there is only one way, and that's going over to this box. And how can you move out of this box? There is only one way. So actually, it's silly to call them probabilities because they are all 1.0 here. But for each of these hidden states, we now have some probability. If you are in this state here, you produce, you could say, or emit, as we say, a symbol that is part of the pattern. And what is the typical symbol of producing or emitting from this first position? Well, the symbols are emitted according to these probabilities here. These are what we call the emission probabilities for the first position, right? And you recognize this is exactly the profile we saw in the previous slide. Exactly the same thing. And from the second position, what are the symbols that are typically produced or emitted here in position number 2? Well, it's a T, right? Because this has the largest probability, but it could also be a C and G, not an A. So each of these hidden states that we call them here will emit a symbol when you visit a state. So you get one symbol from this one, then you move on here, get one from this one, move on, get one from this one, and so on. So you have altogether 8 symbols, right? And if you have a T from the first, a T from the second, a C from the third, an A from the fourth, an A from the fifth, and so on, then you have a very likely sequence, right? That matches this pattern very well. However, if you have an A in the first position and then an A in the second position and then a G in the third position, then you have a sequence that does not match this at all, right? Because it's the wrong symbols at the wrong positions, right? So all the match states, as we call them, that's why they have M in the name, M1, M2, M3, match states. If you have a sequence that matches the pattern, then it should have symbols that correspond to the high probabilities of each of the match states here. So that's our first sort of build the version 1.0 of our hidden Markov model. But actually we haven't done anything other than what we did with this profile, right? But now comes the big trick here. We introduce gaps. Because if we go back to the multiple alignment, and look at, there is one case here. Can you see that? In this position, in position number 3, one of the members, one of the sequences, this one, has no symbol here in position number 3. But it's still a member of this pattern, right? It's still a member of this family. So it is actually possible for a member of this family to have something lacking here in position 3. How can we compensate? How can we sort of include this into our model? So in position 3, one of the sequences had no symbol. And notice this is a minority, right, of the sequences. was lacking something here. But the majority of the sequences has something in position 3. So the normal thing is to have a symbol there. But it could happen that you don't have this symbol in position 3. How can we describe this? And this actually means that this position M3 is not visited when you produce this sequence here, because it doesn't emit a symbol here. Each time you visit M3, you emit something, right? According to the emission probabilities. So it means that usually we go from M2 to M3 to M4 and blah, 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 right? But sometimes we move from M2 directly to M4 without visiting M3. So we sort of go around M3 because no symbol is produced. So how do we actually describe this? Well, the normal thing is to go like this, right? This is the normal path, you could say. Most sequences in this family will go from match state 2, where you produce the second symbol, to match state 3, where you produce the third symbol, to match state 4, where you produce the fourth symbol. But on some cases, on some few cases, we are allowed to go into what we call a delete state. And this one does not produce any symbol. That's the big difference between the delete states and these M states, because all M states produce a symbol, these don't. So a delete state is silent, as we call it. A delete state is introduced when a position usually emits a symbol, but is sometimes silent. So notice that this is the majority way of doing it. This is the minority way of doing it. And that's why this error is slim and this is thick. So when you are here, you now have two ways of going out of this box, right? The most probable path is here, but there is also a possible, however unlikely way of going, which is here. Because only one out of many sequences did this, right? So this is the idea of the delete states. You can go around a match state. Yeah, and typically we then have the probability of going to M3 from M2. That's what this means, the conditional probability. Probability of visiting this box, given that you are in this box, is, for instance, 0.9, a high probability at least. And then the probability of visiting D1, given that you are in M2, is smaller. So these actual numbers here I just took out of the thin air, but these just indicate this probability is always big and this is small. Exactly how big and how small varies, of course, given the data. And then once you are in M3, there is no longer any option. You have to follow the thick arrow. So this is 1.0. And also the same up here. Once you are in D1, there is no other way than going back into the line here, in this case. In reality, there is usually another option here, namely jumping from delete state 1 to the next delete state. So you can actually go around both M3 and M4, right? And then go back into the pattern again. We will see. Okay, so here we have another sort of exception from the rule, right? Notice that in this multiple alignment, and here I just cut out the last part of it, in the 7th position, there are not only a few gaps, but a lot of gaps. Lots of gaps here. And only the minority has some symbol here, but the majority has no symbol in this position. So we also have gaps, but now the roles have been reversed, you could say. The typical is to have a gap here, and the non-typical is to have something emitted. And what do we do then? Then we cannot use the delete state anymore, because a delete state will only be used where we skip a symbol. Here we sort of add symbols. The minority adds some symbols here. So in position 7, only some of the sequences has a symbol. So the normal is to not have a symbol there, you could say. And this means that position-specific emission is not the rule, but rather the exception. And this M7 is not really part of the pattern at all. So we throw it out. We discard it from the model. And then we say that, okay, from M6, instead of going to M7 and then to M8, which is how it looked like in the previous slide, we now sort of replace this by saying that, okay, we go directly from M6 to M8, but sometimes for these first here, right, they have something here in that position. Sometimes we visit an insert state, as we call it. And this insert state will now add a symbol to the sequence. So the normal is to go here and not add anything, but occasionally, reflected by the thin arrows instead of the thick one, occasionally some members of this pattern will have a symbol here, emitted by this one. And then we go back into the pattern again. So an insert state emits symbols, right? They're different from the delete state. They didn't emit symbols, but these ones do. But they emit symbols by what we call background probabilities, which means there is nothing position specific about them. All symbols can occur. Think of it as a flat probability distribution. All the 4 letters here are equally likely to occur. Or if it's amino acids, it's 20 letters. And the insert introduced, we introduce these insert states when extra symbols are sometimes emitted, right? When the minority has something extra. And now we would typically, as I wrote down here, instead of calling this M8, since we sort of took away M7, we would just now rename this and call it M7, right? Because we want to have one match state for each position in the sort of typical pattern. So the typical pattern is not 8 positions long, it's 7. Right? Most of these sequences have 7 symbols, not 8. It looked like 8 because we had these gaps, right? But you have to realize that gaps are not really a part of the sequence, right? That's just something we introduce when we align them. So most of the members of this pattern had 7 symbols. A few of them had 8. So now we have 7 match states. So this is how these models look like in the end then, when we have added, we have the sort of the backbone, you could say, of the model, which is the blue match states in the middle here. But we also have lots of delete states because it is possible that we have some ways of going around each match state if we assume that all match states can be sort of surpassed. And also we can add something extra in between each matching or typical symbol. And then we add also a special state here called the begin state. I call it M0 here now. And then an end state over here, M8. And these are also silent, and this just marks the start and the stop of the whole pattern, right? So you can see we always start here, and then we move most typically along this backbone here and produce 7 symbols, and then you are done, right? You can, however, always sort of divert into different paths here. But these arrows here are the most likely ones. I did not make them thicker in this slide, but think of these arrows as the thick ones and these others as very small, thin ones. They are unlikely, and some of them can even be 0, so we can throw them away entirely. But in principle, we draw it like this. Each M state has its own position-specific emission probabilities. So that's the profile probabilities, right, that we saw. Behind each of these blue ones, there is this new column in that profile. All the insert states, they have identical emission probabilities. So they are not specific, whether you are in this I1 or I2 or I3 or I7, they all produce symbols with exactly the same probabilities. Because there is no, these are just some extra, right? If it had been position specific, it should have been a match state instead, right? That's the whole idea. All the D states are silent. Yeah, they produce nothing. That's the gray boxes here, right? The blue boxes are position specific emissions, while the pink ones are the extra symbols that are also producing something, but with exactly the same probabilities all the time. And then the transition probabilities are all the arrows. And they determine the flexibility here, right? If all but these ones are 0, then there is no flexibility. But as soon as you allow some of the other arrows to be a little bit bigger than 0, then you add some flexibilities of adding or subtracting symbols from your pattern. Okay, so how do we actually, when we have a PHM of this kind, how do we actually search with our sequence? Well, we have a query sequence Q. Could be, in this case, then an ordinary DNA sequence. We have a database of a number of these profile hidden Markov models. And then we take this Q and score against every single PHMM and see how well they score, you could say, and then are listed all the PHMMs that we actually got a good score against. Right? If this sequence Q contains a pattern that matches one of these PHMMs, it gets a high score and is listed as a significant match. So this is the, in principle, how you do it, right? You have a database of these models, you have a sequence, and then you try to score them. And we should now have a look at how this scoring is done, at least so that you have seen it once. Scoring a sequence with a PHMM. So here we have that PHMM again. What we are looking for is finding the most probable path through the model given the sequence. And what do I mean by a path? A path starts here at the beginning, then it follows the arrows in one way or another through the model here in such a way that you get the largest probability if you multiply together these arrows. Think of it as simple as that. The probabilities behind the arrow. If you multiply together the biggest values, you get the biggest score. Right? But of course, so the best thing is always if you can just go through the match states here, right? But then, of course, your sequence also must have the symbols that are the most likely ones emitted from the match. And it must contain exactly 7 symbols, and it must be a T in the first, we remember, a T in the second, and so on. In case your sequence doesn't have this, it deviates a little bit, right? Then you may have to go around or visit some of these other states, but as few as possible, really, right? In order to get a high score. So the Viterbi score, the Viterbis algorithm is what we will look at now. And what is called the Viterbi score is actually the log transform probability of this path. So notice that behind all arrows, there is a probability. So the probability of a path is simply a product of all these arrow probabilities. And if this is as big as possible, the better score we have. So the score is actually this probability, log transform though, but that's just for mathematical convenience actually. And then we can also compute an E-value as we are familiar with from BLAST and so on. But this is just a matter of how large is a large score, right? Then you have to know something about what would you get with the random sequence here. And that's a different, that's more statistical consideration that we will not dig into here. But let's say a few words about how this Viterbi scoring is done. And before we start looking into the algorithm itself, let's go and have a look at sort of the mathematics behind this drawing here, right? Because this is a drawing that you find in all kinds of bioinformatics books and stuff. But what is actually the mathematics behind it? First of all, we need what we call a transition matrix. And what is this? This is simply a matrix with probabilities for each arrow. For each arrow in this drawing. So every single arrow here in this drawing has a value in this matrix. So here, this matrix here is only the upper left corner, right? Because you can see we have only the first states here, right? And the first states here. And it's always symmetrical in the sense that you have all the states listed down in the rows here, and also exactly the same states listed in the columns out here. But it's much bigger than this. So I just listed the first few rows and columns, right? And notice now, when you look at the figure here, we always start in M0. There are 3 arrows going out from M0. This one, and this one. If you go over here, yeah, the probabilities of each arrow is in the model, right? And some of these are 0, right? Because there are no arrows, right? So, for instance, going from M0 to M0, there is no arrow going from M0 to M0. So let me, these were the text I was looking for. So the rows in this matrix, you interpret as coming from. If you are coming from position M0, then you go into some other hidden states, right? You are here in M0 state. Then you can see that there is 3 possible states you can go into from M0. The most likely one is to go into M1. That has the largest probability, right? But it's also possible to go into I0 or D1. But no other. All the other cells are empty or 0 then actually, right? So there are 3 possible ways of going out of M0, and that is exactly the 3 arrows, right? And we can see now from the matrix up here that this arrow is sort of the thick one. And then if you are in I0, then you go down here, you are inside this box. How can you go out of this box? Well, there are 2 ways. You can either follow this arrow and go back to the same box again, or you can go down to M1. And that's exactly what we see here. From I0, you can also go back to I0 with a small probability. And the most likely is that you go into M1. So this is how you read the transition matrix. Once you are in M1, you see that the by far most likely way out of M1 is to go into M2. But you can also go into I1 and D2. And this reflects the 3 arrows. This is the main arrow. These 2 are also possible, but unlikely. And so on. So you can see that every single arrow here has a value in this matrix. That's the transition matrix. We also need an emission matrix, and that is only relevant for these colored boxes. And now I took away all the gray boxes because they don't emit anything. They are silent states. So only the blue and pink boxes can produce a symbol. And also I took away all the arrows because this has nothing to do with the arrows. This is now simply the emission probabilities of the different match states. And notice now we have used this pseudo count, so now there are no zeros anymore, but close to 0 in these 3 boxes and almost 1 here, right? And that's the probability, emission probabilities for match state number 1. And then match state number 2 has its own probabilities in this column, right? and then match state number 3 in the next column, and so on. And then this one here, this is what I call a flat probability distribution. All 4 symbols are equally likely, and this column is the same for all the pink ones. All the pink ones share this one together. So we actually need only to sort of write it once. Because regardless of which pink one you are visiting, this is how the symbols are emitted. You simply draw at random with these probabilities, A, C, G, or T. Anything can occur here if you visit one of the pink ones. So the transition probability for the arrows and the emission probabilities for these colored boxes. That's what we have. You could say that's the mathematical objects we have. And then, yeah, all the silent states, they have no emission, right? So it's 0 by the design. And insertion states have this very peculiar or natural, I would say, property that everything they emit is the same regardless of where you are. Because if I3 had a different emission probability than I4, for instance, then it is sort of position specific. Right? Then this here should not have been an insert state. It should have been a match state then. If there is something peculiar going on here, then it should have been part of the pattern, right? The matches in the pattern, not an insert state. Inserts are extra symbols. So we usually call this E and the transition matrix T, naturally. And then we can start looking at the Viterbis decoding algorithm. And then we need to take a small cup of coffee first, I think. Well, the Viterbis decoding algorithm, let's have a look at it. And the very first thing I should say that if you Google the Viterbis algorithm, you will probably find a very sort of general description of it that can be used for any hidden Markov model. The version I will describe here is very sort of special designed for profile hidden Markov models. Please be aware that profile hidden Markov model that we are looking at now is just a very special type of hidden Markov models. We can use hidden Markov models to describe patterns of all kinds having nothing to do with biological sequences. So this is a special case of the Viterbi decoding algorithm, or it isn't really different from the general one, but it's more sort of specified. It's specified in more detail. So we have a query sequence Q, right? And we wonder now, does this sequence contain the pattern that is described by the profile hidden Markov model, the PHMM? So we also have a PHMM with capital L match states, right? So this is the typical length of the pattern, L. But it could be longer, it could be shorter, right? So we don't really know what to look for, what the size of the pattern is. We know that we always start in a begin state and always end in an end state, right? Begin and end state. So that's match state number 0 and match state number L plus 1, as we saw in the previous slides. We also have the delete states and the insert states. And notice there, we also assume we have the same number, right? L here, or actually L plus 1, because it starts at state number 0. I should mention this delete state 0 is really a strange one. We never visit this one, so it's some sense silly to include it. But I only include it to make the notation complete here, right? Because M's always start at 0, I's always start at 0, and then I like the D's to always start at 0, even if the delete state number 0 is actually never. It's just it's in here just for convenience, you could say. Then a path Y is defined as a sequence of hidden states that we visit leading from begin to end, right? So we know there is a possibility of moving from begin to end through the hidden states. And the path is actually the sequence of hidden states that we visit. So it's not the same as the biological sequence, right? That's observed DNA or protein sequence, but this is something else. This is a series, you could say, of the hidden states that we visit. And the Viterbi algorithm looks for different paths and tries to find the most probable path given the sequence for us. And then the probability of this most probable path is actually the score for this sequence. Right? So given the sequence, how can we go from begin to end in the most probable way? And how probable is it, that path? That's the score we are looking for. In order to do these computations, we start by a trick. We add an empty symbol at the start of our query sequence. A symbol number 0, you could say. And if you remember back to BIN310 or an introductory course in bioinformatics, where we looked at dynamic programming algorithms like Smith-Waterman algorithm or Needleman-Wunsch algorithm for how we can align two sequences. Then we also did a similar trick. We added some empty symbols to the beginning of the sequences. And actually the Viterbi algorithm is also a dynamic programming algorithm. It's in many ways similar to Smith-Waterman and Needleman-Wunsch algorithms. It's the same type. We compute something else, but you will recognize some elements if you remember back to those basic algorithms from introductory course in bioinformatics. Also, let X be the index sequence of our biological sequence Q. And what do I mean by an index sequence? We say that Xj, the element number j in X, is a K if the corresponding symbol in Q is symbol number K in the alphabet. So let me explain this. We have an alphabet, right? In our case, let's say we have the DNA alphabet. So let me just write the symbols here. And usually we write them in alphabetical order, but that's not really required. It can be in any ordering we like, as long as we stick to one ordering. And then we say that this is symbol number 1, this is symbol number 2, this is symbol number 3, and this is symbol number 4. And notice it doesn't really matter which one is symbol number 1, 2, 3, or 4, as long as we are consequent and keep this ordering. And now we will say that if we have now a biological sequence down here, let's say we have a sequence that starts with T, C, A, right? That's how these sequences we looked at previously, they typically started something like this, right? If this is the start of Q, then the beginning of the first 4 symbols in X will be 4, 4, 2, 1. So you can see we just replace every symbol with the index in the alphabet. And of course we should have an empty symbol at the beginning. So let me just indicate an empty symbol by 2 quotes like this with nothing in between. And this has index 0. So here you can see we have the Q sequence and the X sequence. And of course now you realize that these two must be of the same length, right? Both Q and X have n elements, right? They have the same length here now. Yeah, you can number from 0 or number from 1. In this case, it's number from 0. I don't remember why. But anyway, this is Q or X correspondingly. It's just 2 ways of writing the same sequence, right? Now the Viterbi algorithm, what is it that we actually seek? We seek a path Y such that the probability of this path and the sequence together is maximized, right? And this is really important. Maximize, right? We look for the maximum probability, the largest possible probability we can find of having a path together with the sequence. And if you remember back to this Smith-Waterman or Needleman-Wunsch algorithms, we also looked for maximum score, right? The largest possible score we can find by aligning two sequences. So this is exactly the same thing, finding the maximum of something. And this time it's a probability. In order to compute this, we now need some data structures, you could say. We need some matrices or tables. And here's the first one. Capital QM is a matrix with rows and columns. So if you now are in row number I and column number J in this matrix, there is a number there. And what is that number? That's the probability of the most probable path, which is what we are looking for, ending at match state number I and emitting symbol number QJ, right? So we end, this is a sort of, imagine you have started on the path, right? You're looking at the path that ends in match state number I. That's why we are in row number I here. And we have also emitted symbols. So now that we are in this match state, we emit symbol number Q, symbol number J in Q, right? That's why we are in column J here. We have also a similar matrix of exactly the same size, you could say, same number of rows and columns. But this has different numbers. So what if you are in row number I and column number J in this matrix? Then you find that this number is the probability of the most probable path again, but now ending in insert site, insert state number I, and then emitting this symbol. So that's the only difference, right? It ends in an insert instead of ending in a match state. And then you have the third matrix, surprise, surprise, QD of exactly the same size again. And this is again, the number in this row I, column J, is the probability of the most probable path ending in delete state number I. But this does not emit anything, right? So this symbol number QJ must have been emitted already, we say. You end here, and this symbol must already have been emitted by one of these other two. Or, yeah, not necessarily one of these two, but some match state or some insert state. Right. Okay, so we have three different matrices here, all of the same size, you could say, same number of rows and columns. And we have to now fill in the values in these matrices. And we, of course, require that the path we end up with here must, of course, begin in the begin state. All paths begin here. And it must end here in the end state, because all paths end here. And this is regardless of how the actual sequence looks like. So these are some requirements then. So all these three are what we call dynamic programming matrices. And notice they have the same sizes. They have L rows, and this was the number of match states, right? The number of match states. So we have L rows and we have N columns, and this is the length of the sequence, our query sequence, Q. So all have the same number of rows and columns. So look, let's have an example here. This is the sequence we have. This is our Q, right? Small q. And we have this model with 7 match states that we saw previously. And it means that this is a Q, yeah. We add this empty symbol at the beginning, and then there are the DNA letters. The corresponding X is, of course, this one, right? And N is 6, because this is 6 elements. And then here is QM. QM looks like this. Notice we have the match states down here, and we have the sequence out here, right? Empty symbol, T, T, C, A, T. I also just numbered the columns below here. This is not the X sequence. It's written up here. This is just numbering the columns from 0 and up, right? And then we have for QI and QD are exactly similar matrices, except that it says I0 to I7 and D0 to D7, right? In the same, the rows are different, have different row names, but there's the same number of rows and the same number of the columns are exactly the same sequence. And now we need to fill in this table starting in the upper left corner and then fill in row by row and column by column till we have filled it in all the way down here. And this applies to all these three matrices. So all the Q matrices are of this type. We need to compute all values in all three matrices, and the value that we get here in the lower right corner is the one we need from each of these three matrices. We need this final result for all three of them. We will see. That's what we need to compute the score. So how can we start filling in this matrix? Well, there are some things we can immediately fill in. First of all, since we always start with this begin state, we can see that in QM, the very first cell, the probability has to be 1. There is no question. You always start there. Probability 1. The rest of the first row and column of this matrix can also be set to 0. Why is that? Well, if you go down the columns and don't move to the right, it means that you jump to the later match states without emitting anything. Because remember, the first column corresponds to the empty symbol. And that's not really possible. You cannot visit the corresponding match states and not emitting anything. So that's impossible. Also, you cannot move to the in the first row without having 0 probabilities, because you cannot be in the begin state and then start emitting symbols. So you have to sort of move diagonally, you could say, in a sense, out of this first match, M0, in the begin state. Well, in this QI matrix, the first column must be 0. And that's because, remember again, the first column corresponds to this empty symbol. And since all insert states emit something, we cannot emit the empty symbol. So you cannot visit the insert states and not introducing a symbol. So that's why we can fill in zeros in the first column of this one. You can also argue in a similar way that the first row of this one must be 0, because delete states, they are silent, and then you cannot move to the right, you could say, in the first row here, because you cannot emit symbols without having visited some of these other states first. Yeah, so the first delete state can never be sort of visited, really. And then the first row of QI and the first column of QD can also be filled in right away, because these correspond to very peculiar paths. The first is if you go emitting all the symbols from the first emission, so insert state. And that's really strange, isn't it? I mean, if remember that these insert states, they just emit symbols randomly, right? All symbols are equally likely. So if this is the one that matches your sequence, if your sequence is completely random, it certainly doesn't have a pattern, and the corresponding probability will be very low, because these the probabilities of visiting insert sites over and over and over and over again is very small. But this can be filled in directly, and the same here. The first column of QD corresponds to going sort of visiting all the delete states before you visit anything else. And that's also really strange, isn't it? Because then you sort of surpass all the match states. And then you are, if your sequence contains the pattern, then it will never show that, right? Because you visit all these delete states first. So these two can be computed right away, and they will surely get very low scores, both of them. But at least we have them filled in, and then we can start filling in the rest by using the following recursion formula. So typically we would now put this inside a double for loop, where we loop from row 1 up to L and from column 1 up to N, right? Because we have filled in I equals 0 and j equals 0. And then you look at how to fill in row I, column j of this QM. Then you need to go one row back and one column back and see what's in the previous cell, you could say, diagonally up, one step to the left and one step up, and then compute this transition probability here. Visiting mi, given that you were in mi-1, yeah, that's usually quite a big probability here. Visiting mi, given that you previously was in an insert state, yeah, that's also usually quite a big probability. And also visiting mi, given that you are in a delete state. Yeah, so all these three are actually quite large probabilities, so it depends on these ones really, what becomes the maximum. Because you're only interested in the maximum of these three numbers here. These three will be three values, right? And you take the maximum and multiply by the emission probability for symbol number j, xj, symbol xj. And then you get the next value in this matrix. And then you have a similar formula for filling in this qi matrix. And notice there is only two sort of alternatives that you take maximum of here, involving if you come from a match state into an insert state, or if you already was in that insert state and keep staying in that insert state. And this has, if you go back and look at the architecture, there are never arrows going from delete states to an insert state. So if in order to come into an insert state, you must come either from a match or the same insert state. That's why it's only 2 rows here, but here we have 3 rows. There are always 3 ways of coming into a match state. And of course, at the very last one here is the same for the QD then. And again, it's only 2 ways of going into a delete state, either from the previous delete state or from the previous match state, never from an insert state. So, and notice there is no emission probability here because it doesn't emit anything, right? Doesn't emit. So that's why it stays in the same column. You never sort of jump to the right here in a sense. You always stay in the same column. Transition probabilities are the probabilities here that you go from one state to another, right? These are the arrows, right, in our model. And here you have the emission probabilities, the profile. They are here only. Okay, so if you implement this with double for loop and put this in the inner for loop, you can actually compute and fill in these matrices from the upper left corner to the lower right, once you have the initiation from the previous slide. And then the last step is just to go from these are the lower right corners now, these 3 matrices. Notice row L, column N, this is the last cell, you could say. And the last step is just to transit from this into the end state, right? So either from the last match state into the end, or the last inserts into the end, or the last delete into the end. And the maximum of these 3 will give you the score. That's the score for your query sequence. Lower right of each matrix and the last final transition probability of transiting into the end state. So if you try to implement this, it should actually work. and you will get a score for each sequence. And then, of course, in a case where we search with many sequences against the whole PFAM database, this has to be done for every sequence against every model in that database. So it naturally will take some time, but this can be done very quick. So it's not super long after all. Okay, let's have a short look at this code for retrieving all the ORFs from a genome and then do something to this. This is actually the code I'm talking about here that you find in my document. So we will actually go through this step by step here now. So let me just comment this. So I copy this code here, and actually I started to copy, as you can see here. I have made myself a script in my module 7 folder, and I started to fill in. So let me just paste the rest in here. This is how the code looks like in the document. So let's step through it. And this is actually a good idea when you get code like this. It's a sort of a pipeline, right, with this pipe operator. It is usually a good idea to stop and have a look at what happens at each step here. Okay, we start by loading the microsec package. I already did that. And then we read the contigs. So this should be straightforward, reading a FASTA file, right? And then we can see the contigs. There are 182 contigs in this file. And since this is a FASTA object, 2 columns, right? We can click it open, have a look, header and sequence. This we have seen many times now. So the next thing we do is to use this function find ORFs to actually find the ORFs. And instead of the pipeline here now, I just put this into a table here now, TBB. I just want to stop after this step and see what happens. So let me run here, and notice the input is the whole FASTA object, right? The input to this function is a whole FASTA object. And now we can see the output, TBB, is now a table with 538,000 rows and 9 columns. And if we have a look at it, yeah, we can see that this is actually a GFF table, because it has this exact 9 columns that GFF tables should have. Sec ID, that's the name of each config, right? This is taken directly from the header of the contigs. If you look at the contigs here, the header sequences, sorry, the header lines describe every config, right? So they have just been copied in here. This is sec ID, sequence ID. Which sequence is this? And the source, yeah, that's only the name of the function. That's really not interesting. Type is ORF for all, so that's also not interesting at all because it's the same for all. Here are the important ones: start, end, and strand. These are the important ones. The score is also lacking because there is no scoring here. It's just retrieving the ORFs. And also this phase, they can also be ignored actually in this case, but they are here because of this feature format, right? So the start, end, and strand are the important ones, and also possibly the attributes. There is only one attribute here, and that indicates if the ORF is truncated, as we say. So what does that mean? Let's look at the first one here. This one is truncated at the left end. Notice that in most cases it says truncated equals 0, 0. And that indicates it's not truncated at either end. But if there is a 1, either the first or the second digit is a 1, it means it's truncated. So in this case, it means the start is really more upstream than position 3, but this is where we start the config, right? So the config doesn't sort of contain the whole gene. And the same for the second one here. But as soon as you start to get sort of inside the config, nothing is truncated. And then when you come towards the end of the contigs again, you can have truncated at the other end, right? but there was no. Here is the next contig, I suppose, which is also one truncated. So these truncated ORFs are not full ORFs, you could say, from start codon to stop codon. In this case, they have a stop, but you don't see the start because it's sort of outside the start of the contig. But apart from that, we have all the ORFs here, and lots of them, right? And typically we see that many ORFs have the same stop codon, like this one, right? Here we have several, all ending at position 563, but they have different alternative starts. So that's the big ORF table. And the next step now, let's go back and include the next. So we just add the pipe here again, and then we stop here instead. Put this one into a temporary table. So let me just delete everything and run it again. So we run this one, read the contig, and then the two steps. And now we can see that, yeah, here it is. The table is now much smaller. Instead of more than 500,000, it's now down to 250,000. And it's also gotten some more columns, actually. It's still a GFF table because it has these 9 columns that all of them must have, but it's also gotten some extra columns here. We can ignore them if we like, or have a look at them. But the point is now, we only now have what we call the LORFs, the longest ORFs. So now every start and stop should be sort of unique here. There are no alternative starts for every stop now, because the LORF is simply for each stop codon, you pick the most upstream start codon. So you have the longest possible ORF given the end or the stop codon. Note, however, that end is not always a stop, right? Because it depends on the strand. So if you are on a positive strand like here, then end is actually the stop codon. But if you are on a negative strand like we are here, then actually the stop codon is here at the start, because then it's on the reverse strand, right? So you should actually read from right towards left. And this I find a little bit confusing. I mean, it's not confusing. The rule is straightforward, but these columns should not have been called start and end, because they are not. They are only start and end on the positive strand, but on the negative strand, it's reversed. So the end is actually the start, and the start is the end, because it's read in the opposite direction, right? So they should have had different names, but okay. We just have to get used to it. So LORF will now only collect the longest ORF, so we have actually unique starts and stops now. And then the next step is to compute the lengths. So let's stop here as well. And notice there is a function called ORF length. It takes the ORF table as input. And since we have it as input through the pipe here, we just use the dot to indicate that it's the table that we have, that we are mutating, that we are computing the ORF length. If we look at the help file for this one, question mark and the name of the function, it's always a good idea if you want to understand how a function works. Length of ORFs. And we can see that length, the first argument is an ORF table, and the second one is just a logical AA for indicating if the length should be computed as the number of amino acids or not. So if this is true, then you get the length of the ORF as the number of amino acids after translation. If you turn it off, you will have the number of bases. Right? So you will have a different value depending on whether you measure the length of a gene in how many bases it has or how many amino acids it will get after translation. And it's basically 3 times as many bases, right? Except that the stop codon is also never translated, so it's the number of bases minus 3 divided by 3, something like that. So now we compute the length as amino acids. So keep this in mind. Do you want the length as the number of amino acids, or do you want it as the number of bases? That's what you can turn on and off here. Let's run this part of the code again. We don't need to read the config yet again. We have it. So now we run the 3 first lines here, and now the table, yeah, it's the same number of rows, of course, but because that hasn't changed. But the length column has now been changed to contain the number of amino acids. And this could, in some cases, now be not an integer. And that's really strange, isn't it? It should always be an integer. No, because in some cases we have this truncated ORFs, right? Where we don't have the full ORF from start codon to stop codon. And here you can see one. This is not a full integer number of amino acids because the ORF has been truncated at the end, we can see here, since it has first a 0 and then a 1. So it's the end codon that is lacking here, stop codon. And this is not a complete ORF, so it doesn't have integer number of amino acids. Yeah. Usually we throw away these truncated ORFs, right? So then all the rest should be integers. So this is now the length in amino acids. Okay, and then we just filter by length here, right? So now we just filter this table and say, okay, only keep the rows where length is at least 78. At least 78. So let's see how it looks like after this step. We go back and run it again. And now the table, yes, look at this. Now it was really shrinked a lot, down to 7,614 rows only. Because now we throw away all these very short, or everything that's shorter than 78, right? And then we are only left with the long ones. So now if you scroll down the length column, you should find no value smaller than 78. You can see the smallest ones are 78, right? Because we used larger than or equal to. So every one of length 78 or more. And then finally, we just filter out these truncated ones. We say that keep only the cases where in attributes this says truncated equals 0, 0, not truncated. We have both the start and the stop codon inside here, and then we are happy. So now if we run it, I guess that will, yeah, a few less, right? 7,500. So there were still a few truncated that we now discarded. So that's our final ORF table here. And now this is a GFF table, even if it has some extra columns. It's still a GFF table. And now we can use this to do the further processing. Okay, so let's have a look at how we can run this shell script directly in the terminal without using the Slurm queue. And this, of course, is something we rarely do, but this time I will do it just to demonstrate, because what we do here is such a small operation that we can actually do it on the login server. Otherwise, that's forbidden, right? But tiny small operations we can do. So here is the shell script, and this is just copied directly from the module 7 document here, right? I have made a text file and pasted in and saved it under the name here, and remember the extension .sh. So what is actually going on in this small shell script? Let's have a look at it. First, we load the Hammer software. Then we set a variable here with the address to the PFAM-A database that we are going to fetch from. We also want to output to a folder here on the scratch called hammer. And we want to create a database, a PHMM database in there that I want to call abr.hm. That's a good name, right? And then in case this outdir does not exist, we create it here, right, with the if statement. And then we use from the Hammer software two commands. First, HMM fetch. It needs these arguments here. Let's start at the very end. First of all, a file of accession numbers. So this file must exist in my folder here now, and I think it does. Yeah, there it is. You can see it down there in my module 7 folder. So this is the results of some previous operations, right? So this file must exist. This is I can click it open just to look at it. It's a small text file with 6 accession numbers from the PFAM database. These are the models I want to retrieve now and put or fetch and then put them into a separate database. So I fetch these accessions number from this database, the PFAM database, and put it into this database that I'm named up here, right? So this is my own personal database. And then in the second statement, we do what we call compressing the database, HMM press. So in order for this to work, you need to have done this first. So you create this file here. You can see that it becomes a file, right? Okay. Let me now move over to the terminal window and list the content of this module 7 folder. And here I can see, first of all, I can see my shell script that we just looked into. Here it is listed. I can also see this file with the accession numbers. It's in the same folder. This looks fine. If I also look into my Scratch folder, like this, we can see it doesn't contain very much. It certainly contains no folder called hammer, right? So we want to output into this. So it means that these lines here need to be executed to create that folder actually first. Or we can create it here and now, but let me just rely on the script doing its job to create that folder here before we start putting things into it. So how do I actually run this shell script now without using the sbatch to the queue? Well, to run a shell script that is in this particular folder, I write just like this. /, and remember dot means this folder. So the first 2 characters here only tells Unix to look into this folder, right? In this folder / the name of the file that you want to execute. HMM fetch, wasn't it? .sh. Yeah, that looks fine. And then return. And then you will typically see this one, permission denied. I was not permitted to do this. So what does it mean? Let's have a look up here again on this line here. If you look at the very left side of this output, you see the permissions. There is a dash first, but that doesn't mean anything. But afterwards, there are actually 9 characters here, those that I highlight here now, 9 characters. And these 9 characters describe the permissions for this file here. Right? You can see all rows have similar. And it's the first 3 characters that tells you your permissions. The owner of the file, the user of the file, has these permissions. And it says R for read. I am allowed to read this file. W for write. So I'm also allowed to write into this file. And otherwise I would not be able to edit it over here, right? So these 2 are always there. These 2 permission files that you own are always you have read and write permissions to them. Oops. But the last one is a dash. So it means it's not set. And this is the execution permission. So that's the reason why we got this permission denied. We are not allowed to execute this file. And that's what we try to do now. We try to execute it. So how can we actually get execution permission for a file? Well, in order to change the permissions of a file or a folder, you use this command, chmod, change mode. So, and then after a space there, you need to specify what permissions do you want to change and for which type of user. And in our case, we type U for user, which is me, right? Or when you type it, it's you. So I want to change my permissions to this file. And what do I want to change? I want to add execute permission. And then another space and the name of the file that I want to change permissions to. So now I need to type in the name of the file here. And like that. So if I now list again the content of this folder, you will notice now that first of all the color changed, but that will vary depending on your terminal window. So don't be too hung up on that. What you should look for is this. Now you can see I as a user have both read, write, and execute permissions. That's all possible permissions to this file. Let me just briefly mention what are the rest of the remaining 6 characters here. Well, the next 3 characters are read, write, and execute permissions for all users in your group. It's possible to group users together in Unix systems so that you can share files within the group, but not outside the group, right? The last 3 characters are permissions for all other users. So if you want to really open your files to all other users, you should change these permissions. But I would not recommend, at least not give writing permissions to other users, because then anyone can change your files, right? That's really stupid. It could be sensible in some cases to give execute rights to other people, because you want them also to execute your files. And how would I do that? Then you would type O for others instead of U for users, and plus X in this way. And the name of the file, right? So if I now look at the content again, you will see I now open this shell script also for everyone else to run. But not write, not the right permission. But okay, the last one here is not really needed, right? You only need to set the execution for yourself. And now we can try to run this script again. I just used the up arrow up here, yep. And then let's see if it's running. Yep, it looks like it worked fine. And gave some output. And now we can have a look into my scratch disk. I should do that now. Scratch disk again to see now it has been created a folder called hammer there. And if I just look into that folder as well, yes, we will see this file was created by the HMM fetch statement, the first one here. And then all the other files were created by the second HMM press statement. So now I actually have my own personal little tiny PHMM database here consisting of 6 different models. that I can use for searching against, just like we searched against the PFAM-A database.